Hello everyone, welcome to the next lecture in our physiology lecture video series. This lecture is titled The Heart, and in this lecture we will learn about the structure and function of the heart, how exactly the heart is divided into separate chambers which act independently of each other, and how the heart is able to achieve the pumping of blood throughout our circulatory system. This particular lecture will refer back to the previous lectures, which were the nerves and the muscles, as we will talk about the pumping of the heart in terms of muscle contraction. And I will urge you to look back to the previous videos on the channel in case you do not understand anything about the things that we will refer back to. So without further ado, let us begin. To start off, let us look at the structure and function of the heart. This is a schematic showing the heart, and as we can see right here, the heart is divided into two parts: the right heart that pumps blood through the lungs, and the left heart that pumps blood through the peripheral organs. So as we can see, this is the schema that we will follow throughout the lectures, in which oxygenated blood is shown in red and deoxygenated blood coming from the body is shown in blue. This is the right part of the heart, and you can imagine the heart as the you can imagine the schematic as one where you are standing on the other side of the screen. So the right and the left halves are flipped. So this is the left half, and this is the right half of the heart. The right half, the right half of the heart takes blood incoming from the body, which is deoxygenated and hence is shown in blue, pumps the blood back, pumps the blood through it into the lungs. The oxygenated blood from the lungs come back into the left heart, from which it is pumped out into the whole body via the aorta. Each of these hearts is a pulsatile two-chamber pump composed of atrium and ventricle. So essentially, the heart has four chambers: the right atrium, right ventricle, left atrium, and left ventricle. Each part of the heart is divided into two parts: the atrium and the ventricle. The atrium is a weak primer pump for the ventricle, which helps to pump blood into the ventricle, and the ventricle is the one which actually pumps it through parts of the body. The ventricle, as we have said, is the main supply pumping force that propels the blood either through the pulmonary circulation by the right ventricle. Pulmonary means the circulation that goes to the heart. So the right ventricle is responsible for pulmonary circulation through the pulmonary artery, or through the peripheral circulation by the left ventricle. And the left ventricle is the one that pumps the blood into the body via the aorta. Next, let us go into the valves that represent in the heart. There are four valves that are present in the heart that we will go through. The first is the mitral valve that lies between the left atrium and the left ventricle, which is present right here. The next valve is the tricuspid valve, which lies between the right atrium and the right ventricle. The pulmonary valve is present at the junction of the right ventricle and the pulmonary artery right here. And the aortic valve is present at the junction of the left ventricle and the aorta, which is present right here. And these valves help in unidirectional flow of blood through the heart, so that the blood does not pump back in the opposite direction into previous chambers and flows accordingly to where it is needed. Such that in case of the mitral valve, obviously the mitral valve is unidirectional and allows flow of blood only from the left atrium to the left ventricle. And if the ventricular blood tries to flow back into the left atrium, then the valve closes and prevents the backflow. Now we will talk about the cardiac muscle. So cardiac muscle is the third type of muscle that is present in our body and is different from the skeletal and the smooth muscles that we have already discussed in the previous lecture. The heart is composed of three major types of cardiac muscle: the atrial muscle, the ventricular muscle, and specialized excitatory and conductive muscle fibers. The atrial and ventricular muscle contract like the skeletal muscle. There are many similarities between them, but the cardiac muscle contract for much longer, which means that they can maintain the strength of contraction much longer than that of the skeletal muscles. Conversely, the specialized excitatory and conductive fibers that are the third muscle type present in the cardiac muscle do not actually take part in contraction or pumping of the blood. Instead, they exhibit automatic rhythmical electric discharge in the form of action potential or conduct action potentials across the heart. So what these specialized fibers do is that they produce the rhythmicity of the heart. The rate at which the heart beats is directed by these specialized fibers. These fibers are also responsible for conducting the action potential or the depolarization wave throughout the whole heart. The physiology of the heart muscles can be described as follows. Cardiac muscles, as skeletal muscles, is striated in nature. It also has typical myofibrils that are present in all muscles, which are the actin and myosin filaments. These filaments slide along each other for contraction via the sliding mechanism that we have already seen. So the mechanism of action is not very different, but as we will see, the excitatory coupling of the cardiac muscles are very different from that of normal skeletal or smooth muscles, which allow them to act as they do, like allows them to maintain the strength of contraction for way longer, and we will see how they do it. But first, let us go through the necessary topic, which is cardiac muscles act as a syncytium. A syncytium is a particular object or a system that works as one. So the cardiac muscles do not work individually, but when the heart beats, it beats as one. Cardiac muscle fibers are made up of individual cells connected in series or in parallel with others. So like this, they are either present in series or parallel to each other. At each there are specific intercalated discs present, which are shown in darker color right here, via which the cell membranes are fused to one another, such that they form permeable communicating junctions or gap junctions. So what are these gap junctions? Gap junctions are essentially particular protein pores present within the cells, so that the internal environment of the cells can be continuous. Thus, when a depolarization wave travels from the uppermost cells, the flow of ions can easily travel to the next cells via these gap junctions. And these gap junctions allow almost totally free diffusion of ions, such that the action potential can easily travel from one cardiac muscle cell to the other. And thus, when one cardiac muscle cell exhibits action potential, all others can easily take up the wave of depolarization and commit to the action potential as well. The heart is comprised of two independent syncytiums, namely the atrial and the ventricular syncytium, which contract independently of each other. The AV bundle is a specialized fiber tract that connect the two syncytiums, the atrial and the ventricular parts. We have to know the one important point is that the atrial and the ventricles are divided from one another by insulatory muscles, which prevent the flow of the action potential from any other part other than the AV bundle, which connects the two. Next, we move on to action potential in cardiac muscles. We'll look in detail into the action potential itself. The action potential recorded in ventricular muscle fiber averages about 105 millivolts, meaning that from the lowest point to the peak of the action potential, the magnitude is 105 millivolts. The intracellular potential rises from -85 millivolts to about +20 millivolts during each beat. which is very common and similar to normal nerve fibers and skeletal muscles. After the initial spike that we can see right here, the membrane remains depolarized for 0.2 seconds. And we see a plateau-like appearance that appears just as this. If you remember the nerve action potential or the skeletal action potential, the action potential used to rise like this to the peak and fall down sharply. But instead, in the cardiac muscles, we see a plateau-like appearance. And thus they exhibit a plateau followed by, at the end of the plateau, by abrupt repolarization. So at the end of the 0.2 seconds, at the end of the plateau, their potential drops abruptly down to repolarization levels. The presence of this plateau in action potential causes the ventricular contraction to last as much as 15 times as long as in cardiac muscles compared to skeletal muscles. So this plateau allows the cardiac muscle to remain contracted for a much longer time than skeletal muscles in response to one action potential. This, the top part, gives us a representation of specialized Purkinje fibers, which are present in the ventricles and help in conduction of the action potential. And we are shown the membrane potential, the Purkinje fibers, in red in the top, and in blue in the bottom. We can see the action potential as it is exhibited by the ventricular muscle itself. And as we can see, ventricular muscle also shows a plateau-like appearance. Next, we will look into how exactly cardiac muscles are different from skeletal muscles, which causes them to have a longer period of contraction, and what exactly is the reason for this difference. The difference between the cardiac and the skeletal muscle arises from the fact that the action potential of the skeletal muscle is caused by the opening of large number of fast sodium channels, as we had seen in the previous video. While in the cardiac muscles, the action potential is caused by opening two types of channels. The same fast sodium channel as is present in the skeletal muscle, and a different population of slow calcium channels, which are slower to open and remain open for a much longer time than the sodium channels. Further, immediately after the onset of the action potential, the permeability of the cardiac muscle membrane for potassium ions decreases about 5-fold. This greatly decreases the outflux of potassium ions and thereby prevents early depolarization. So, if you remember the previous videos, you will know that after an action potential is initiated, once the membrane depolarizes completely and reaches the peak, repolarization occurs by opening of potassium ion channels, which allow flow of potassium ions out of the cell. The flow of potassium ions out of the cell now causes the interior of the cell to become negative as positive ions are flowing out, and this begins the process of repolarization, by which the membrane potential again drops back to the original resting levels. But in cardiac muscles, as the action potential is initiated, the permeability of potassium ions through the muscle membrane decreases about 5-fold. This means what this means is that the repolarization process is hindered, and thus it takes a much longer time to repolarize the membrane. That is, allow the membrane to drop down back to the minus 85 millivolt level, and thus we see the plateau. This, coupled with the fact that the calcium channels, which allow the entry of calcium ions into the cell, which maintain depolarization, the calcium channels which maintain depolarization are open for a much longer time. Both of these factors combined give rise to the plateau-like appearance of the action potential in cardiac muscles. Further, there is another point of difference between skeletal muscle and cardiac muscle, that is the velocity of conduction of excitatory action potentials. Action potentials travel through the atrial and the ventricular muscle fibers at about 0.3 to 0.5 meter per second, which is 250 1 by 250 times lower in velocity as compared to very large muscle fibers or 1/10 of the velocity in skeletal muscle fibers. Thus, the impulse also travels very slowly through the cardiac muscles as compared to the skeletal muscle fibers. Next, we move on to the concept of refractory period in cardiac muscles. Refractory period, as we know, is the period during which a cardiac muscle cannot undergo action, cannot exhibit a action potential again. So once the cardiac muscle has an action potential, it needs to rest, which during which it repolarizes its membrane, after only which the another action potential can be initiated. So there is a gap between consecutive action potential, and this is known as the refractory period. The normal refractory period of the ventricle is 0.25 to 0.30 seconds, which is about the duration of the prolonged plateau action potential. So till the plateau is there and the membrane potential drops back to the initial resting levels, we will not be able to reinitiate an action potential, and this is the refractory period, which is shown right here. There is another additional relative refractory of about 0.05 seconds, during which the muscle is more difficult than normal to excite, but nevertheless can be excited by a very strong excitatory signal, which is demonstrated by the early premature contraction shown right here. And finally, we also can see a late premature contraction, which is after the refractory period and exciting the cardiac muscle before its normal beating pattern, but after the refractory period can allow it to undergo another action potential, as is shown by the later premature contraction. This period of 0.25 seconds after which the action potential can be generated is known as the absolute refractory period. In this period, no action potential can be generated in the cardiac muscle. But the relative refractory period, as we have seen, is another 0.05 seconds after that, during which early premature contraction or action potential can be generated, but it requires a very high excitatory signal. Next, we move on to the excitation contraction coupling in cardiac muscles. Excitation contraction coupling is the mechanism by which action potential causes the myofibrils to contract. And in this particular discussion, we will know how exactly the cardiac muscles are different from skeletal muscles on the molecular level. As in the skeletal muscles, there are similar mechanisms in place, such as release of calcium from sarcoplasmic reticulum and spread of action potential by transverse tubules. But the release of calcium allows interaction between actin and myosin, which allow the muscle contraction. So these parts are the same as we have seen in skeletal muscles. However, there are various points of difference that we will see. The cardiac muscle has a difference in which the calcium coming from the sarcoplasmic reticulum is not entirely sufficient to allow the cardiac muscles to contract, and hence a large amount of extracellular calcium diffuses in from the T tubules or the transverse tubules themselves. That is, the extracellular fluid contributes calcium ions for the muscle contraction. This extracellular calcium is critical for cardiac muscle contraction, as the sarcoplasmic reticulum is less elaborate and does not store enough calcium. And thus, the extracellular calcium is essential for cardiac muscles to function properly. Also, inside the transverse tubules, there is a large quantity of mucopolysaccharides that are electronegatively charged, and these store abundant amounts of calcium, which are then kept available for diffusion into the cardiac muscle fibers. So the transverse tubules are an abundant store of calcium ions, where calcium ions are stored via these mucopolysaccharides, and when required, the calcium ions are allowed to diffuse into the cardiac muscles and allow the cardiac muscles to contract. At the end of the plateau of the cardiac action potential, that is one contraction, the influx of calcium ions into muscle fibers is suddenly cut off, and the calcium ions in the sarcoplasm are rapidly pumped out of the muscle fibers into the sarcoplasmic reticulum and the transverse tubule and the extracellular fluid space. The calcium being pumped out is essential to prevent, to stop the contraction of the heart muscle and allow it to relax. As a result, the contraction ceases until a new action potential comes on and again allows the muscles to contract, which will require the calcium to flow back into the sarcoplasm. The duration of the contraction in atrial muscles is 0.2 seconds and in ventricular muscles is 0.3 seconds. Next, we'll look into the cardiac cycle itself. The cardiac events that occur from the beginning of one heartbeat to the beginning of the next are called the cardiac cycle, which is demonstrated by this picture right here. Each cycle is initiated by spontaneous generation of action potential in the sinus node. The sinus node is one of the specialized excitatory fibers that we were talking about. And it is the pacemaker of the heart, as we will see in later slides. Once the spontaneous generation of action potential is done by the sinus node, the action potential travels from here through the atria, then through the AV bundle into the ventricles. Thus, this traveling of the action potential from the atria to ventricles causes a delay of more than 0.1 seconds to occur while the action potential travels from the atria to ventricles, and thus the atria can contract ahead of the ventricles. This is necessary because the atria pump blood into the ventricles before the ventricles contract, and thus atria should start contracting before the ventricles. The contraction of the atria pumps blood into the ventricles before the strong ventricular contraction begins, which pumps blood throughout the body. Next, we'll go through the concept of systole and diastole. The cardiac cycle consists of a period of relaxation during which the heart rests, which is known as the diastole, during which the heart fills with blood. This is followed by a period of contraction called the systole. So the systole and the diastoles are shown as these lines right here. And we can see as the systole begins, the pressure rises, and at the end of the diastole, the pressure falls down. The figure shows the different events that occur during the cardiac cycle for the left side of the heart. The top three curves show the pressure changes in the aorta, the left ventricle, and the left atrium respectively. So the first dotted line is the pressure change in the aorta, followed by the red line, which is the pressure change in the ventricle, and finally the lower dotted line, which shows the pressure changes in the left atrium. The fourth curve depicts the left ventricular volume, the fifth electrocardiogram, and the sixth is a phonocardiogram, which is the recording of sounds produced by the heart, mainly by the heart valves as it pumps. The electrocardiogram is an important medical test that is performed to check if one's heart is working perfectly. It consists of the P, Q, R, S, and T waves, which are shown right here. These are actually electrical voltages generated by the heart and recorded by an electrocardiograph from the surface of the body. The P wave that is shown here is the spread of depolarization through the atrium, followed by atrial contraction, which causes slight rise in pressure in atrial pressure. About 0.16 seconds after the onset of the P wave, we have the Q, R, and S wave occurring, which is a result of electrical depolarization of the ventricles. And finally, we observe the T wave, which represents the repolarization of the ventricles when the ventricular muscle finally begins to relax. And then thus are now prepared for the next cycle of contraction. So we will go through the functions of the atrium. The atria is a primer pump which transfers blood into the ventricles. Blood normally flows from the great veins into the atria, and about 80% of the blood flows directly through the atria to the ventricles even before the atria contracts. So this flow of blood is passive and just passes right through the atria into the ventricles. Then atrial contraction occurs, which causes an additional 20% filling of the ventricles. Therefore, atria simply act as primer pumps which increase the ventricular efficiency by about 20%. However, this atrial pumping is not really required, as the 20% extra effectiveness is nothing much compared to the capability of the heart to pump 300 to 400% blood than is required by the resting body. So the heart is capable of pumping 300 to 400% more blood than is normally required, and thus the 20% effectiveness, increased effectiveness by the atria does not contribute a lot to the heart's overall efficiency. The pressure changes of the atria are given by the A, C, and V waves. Let us go through the A, C, and V atrial pressure waves one by one. So this is the lower dotted line is the atrial pressure curve, and here we can see the first is the A wave. A wave is caused by the atrial contraction, which pumps the extra 20% blood into the ventricles. This is followed by the C wave. The C wave is the rise in pressure in the atria, but it is not due to contraction of atrial muscles, but instead it is due to the contraction of ventricular muscles. When the ventricles begin to contract, the force of the ventricular contraction pushes blood back, some blood back into the atria. This blood flow shuts off the AV valve that is present between the ventricle and the atria, but this increases the pressure in the atria itself, and thus we see the C wave. And finally, we have the V wave at the end of the ventricular contraction. It is due to the flow of blood into the atria from the great veins, while the AV valve is still closed due to ventricular contraction. So this was these were the A, C, and V waves. Next, we move on to the function of ventricles. The first function is the first thing we will look into is how the ventricles are filled. As the ventricles relax during ventricular systole, large quantities of blood accumulates passively into the right and the left atria due to closed AV valves. So after the ventricular systole, the valves are still closed because there is still pressure inside the ventricles, and this allows accumulation of blood in the atria. As soon as the systole is over and the ventricular pressure falls again to the low diastolic values, the moderately increased pressure that have developed in the atria during the ventricular systole immediately pushes the AV valves open. As we know that the AV valves are unidirectional, and as the pressure is slightly greater on the atrial side during ventricular diastole, the AV valves open and allow the entry of blood into the ventricles. And this opening allows blood to flow rapidly into the ventricles, as shown in the figure. So this is the this is where when the AV valves open, and this is where the AV valves open and allow the flow of blood into the ventricles. This period is known as the period of rapid filling of ventricles, which is shown right here. The period of rapid filling lasts for about the first third of the diastole. This is the first third of the diastole. During the middle third of the diastole, a small amount of blood normally flows into ventricles. This is the blood that continually empties into the atria from the veins and passes directly through the atria into the ventricles. During the last third of the diastole, the atria finally contract, which is the A wave, and give an additional thrust to the inflow of blood into the ventricles. This accounts for about 20% of the filling of the ventricles during each heart cycle. So first we have in the first 1/3 of the diastole, we have a feeling of, we have accumulated blood in the atria falling directly into the ventricles, followed by a direct flow of blood from the veins via the atria into the ventricles, and finally atrial contraction, which pumps extra 20% blood into the ventricles. Next, we have the emptying of ventricles during systole. This emptying of ventricles is divided into two parts: the period of isovolumic contraction and period of ejection. Immediately after ventricular contraction begins, the ventricular pressures rise abruptly. So this is the rise in ventricular pressure, which causes the AV valve to close. Here the AV valve starts closing. Then an additional 0.2 to 0.03 seconds is required for the ventricles to build up sufficient pressure to push the semilunar, which are the aortic and the pulmonary valves, to open against the pressures in the aorta and the pulmonary artery. So the aorta and the pulmonary artery are kept at our muscular vessels of blood and have a pressure of their own. Normal aortic pressure is close to 80 millimeters of mercury, and thus the ventricle, in order to open the semilunar valves, need to cross this 80 millimeter of mercury threshold. And this and while this sufficient pressure is being built in the ventricles itself, there is contraction occurring, but there is no emptying of the ventricles. This is because the AV valve has already closed and aortic valve is about to open, so the blood has nowhere to go, and thus there is no change in volume of the ventricles, but the tension or the contraction in the muscles is increasing. This period is known as isovolumic or isometric contraction, meaning that the tension is increasing in the muscles, but little or no shortening of muscle fibers is occurring. This is the period of this is the part of the plot which shows the isovolumic contraction. We see that there is very little change in the volume, but a huge increase in pressure. Next, we have period of ejection. When the left ventricular pressure rises slightly above 80 millimeters of mercury, which is just crosses the threshold of the aorta, and the right ventricular pressure slightly above the 8 millimeter mercury mark, which is the threshold for the pulmonary artery, the ventricular pressures push the semilunar valves open. Immediately, the blood begins to pour out of the ventricles, with about 70% of the blood emptying during the first third of the period of ejection. So during this time only, 70% of the blood is being ejected, and remaining 30% is emptied from the ventricles during the next 2/3 during the period of ejection. Therefore, the first third is known as period of rapid ejection, and the last 2/3 are known as period of slow ejection. Next, we move on to period of isovolumic relaxation. So the previous was isovolumic contraction. This is isovolumic relaxation. At the end of the systole, the ventricle relaxes, and this allows the right and left intraventricular pressures decrease rapidly. So this is the fall that you can see right here. The elevated pressures in the distended large arteries that have just been filled with the blood from the ventricles now push back and try to backflow into the ventricles. But this backflow allows the aortic and the pulmonary valves to snap close and prevent blood to flow back. For another 0.03 and 0.06 seconds, the ventricular muscles continue to relax, even though ventricular volume does not change. As there is no flow of blood anywhere, ventricular volume does not change, but the muscles are contracting. And this gives rise to the period of isovolumic or isometric relaxation. During this period, the intraventricular pressures decrease rapidly back to their low diastolic levels. So we again reach the starting point of the previous cycle. and then the AV valves again open to begin the new cycle of ventricular pump. So the AV valves will open again, the atria will pump blood back into the ventricles, and the whole cycle will repeat. Next, we go into various parameters that are used to judge the effective pumping by the heart. These are the end diastolic and systolic volumes as well as the stroke volume output. During diastole, normal filling of the ventricle increases the volume of each ventricle to about 110 to 120 milliliters. This volume is known as the end diastolic volume. So at the end of the diastole, the amount of volume present in the volume of blood present in the ventricles is known as the end diastolic volume. As the ventricles empty during systole, the volume decreases by 70 milliliters. And this emptied volume, or the amount of blood that is pumped out, is known as the stroke volume output. The remaining volume in each ventricle is about 40 to 50 milliliters. This blood is the one that was not pumped out and remains in the ventricle even after systole, and is known as the end systolic volume. The fraction of end diastolic volume that is ejected is known as the ejection fraction. So the ratio of end diastolic volume to the amount of blood that was ejected is known as the ejection fraction, and is usually about 60%. When the heart contracts strongly, as during exercise, the end systolic volume can decrease to as little as 10 to 20 milliliters of blood. So from 40 to 50, we have a decrease to 10 to 20 milliliters when the heart contracts strong. When large amounts of blood flow back into the ventricles during diastole, the ventricular end diastolic volume can become as great as 150 to 180 milliliter in a healthy heart. Thus, by increasing both the end diastolic volume and decreasing the end systolic volume, the stroke volume output can be increased to more than double normal. So that brings us to a close on discussion on the cardiac cycle, and we move to discuss more about the cardiac valves. So there are, as we have seen, two valves: the atrioventricular valves present between the junction of the atria and the ventricles, and the aortic and pulmonary valves that guard the junctions between the ventricles and the arteries that they pump blood into. The AV valves, the tricuspid and mitral valves, prevent backflow of blood from the ventricles back into the atria during systole. The atrioventricular valves open and close passively due to pressure exerted by the blood. That is, they close when a backward pressure gradient pushes blood backward, and they open when a forward pressure gradient forces blood in the forward direction. So these AV valves can be thought of as a one-way door. When the door is being pushed from the correct side, the door opens, but when it is pushed from the opposite side, the pressure of the blood actually closes the door or the valve. And this is how they work passively. So there is passive diffusion of blood that actually guides their opening and closure. These AV valves are further supported by chordae tendineae or papillary muscles, which are bound to the heart walls and help in opening and closure of the AV valves. The aortic and the pulmonary valves, on the other hand, are known as semilunar valves. The high pressures in the arteries at the end of systole causes the semilunar valves to snap close, and which is in contrast to the much softer closure of the AV valves. Secondly, there is another point of difference between AV valves and semilunar valves, which is because of the smaller opening of the aortic and the pulmonary artery valves, velocity of blood ejection through the aortic and pulmonary valves is far greater than that through the much larger AV valves. So the semilunar valves, the blood passing through the semilunar valves goes through at a much higher velocity than blood that passes through the AV valves. And finally, because of the rapid closure and rapid ejection of blood, the edges of these valves are faced with greater mechanical abrasion as compared to the AV valves. And further, they are not supported by any papillary muscles or muscles that connect to the heart wall, and thus only are supported by the connecting points between the heart muscles and the valves themselves. Next, we move on to the volume pressure diagram and cardiac workout. The volume pressure diagram, as is shown here, is the plot of volume versus pressure that occurs in the left ventricle during the cardiac cycle. This whole volume pressure diagram is divided into 4 phases. The phase 1 is the period of filling, where the ventricular volume is about 45 milliliters after the previous heartbeat, and it is obviously the end systolic volume. As the venous blood flows back into the ventricle from the left atrium, the ventricular volume rises slowly and reaches the point that is shown here, where the ventricular volume has reached 115 milliliters of blood, which is the end diastolic volume. This brings us to the second phase. The second phase is the period of isovolumic contraction. The volume of the ventricles does not change, and as we see, it is a vertical line parallel to the y-axis, which means is that the volume of the ventricles has not changed. Instead, the pressure has only increased from this point to this point. The volume of the ventricle does not change because all valves are closed. The AV valves as well as the semilunar valves are all closed. However, the pressure inside the ventricles increases to equal the pressure of the aorta at a pressure value of around 80 millimeters mercury. So the pressure only increases in the ventricles, which finally causes the opening of the aorta, the opening of the aortic valve. Opening of the aortic valve brings us to the third phase in the cardiac cycle, which is the period of ejection. During ejection, the systolic pressure rises even higher because of still more contraction of the ventricle. At the same time, the volume of the ventricle decreases because blood has started to flow into the through the aortic valve into the aorta, and thus volume of the blood also decreases. So we have a movement along the towards the negative side of the x-axis, which indicates the ejection of blood, while the pressure still increases. And finally, we come to this point, which is the phase 4, where which is the period of isovolumic relaxation. At the end of the period of ejection, the aortic valve again closes, this time due to the backflow of blood from the aorta, and the ventricular pressure falls back to diastolic pressure levels as the ventricles relax. Thus, the ventricles return to their original starting point of 45 milliliters of blood left in the ventricles, and around this point there is a 0 millimeters of mercury pressure in the atrium. So we return back to the original position that we started from. This cycle continues till throughout a person's lifetime. The area subtended by the volume pressure curve, that is the yellow shaded area that we can see right here, gives us the net work output of the ventricle during the cardiac contraction cycle. And the bluish lines that we see overall that are here are actually the maximum contraction and the volume of blood that can be flown into the ventricles artificially. And thus we come to an end to our lecture today. In this lecture we learned about the structure and the functioning of the heart, and the references are given right here. In the next lecture we will learn exactly how the action potential travels through the heart itself, and look closely into the flow of action potential from the sinus node, and as well as the control of the heartbeat via the nervous system as well. Thank you for listening to this video.